We previously discussed the possibility of using mesenchymal stem cells for the treatment of type 2 diabetes because these cells have, number one, anti-inflammatory properties, which hypothetically can decrease insulin resistance. Number two, mesenchymal stem cells appear to have the ability to regenerate islets or become insulin-producing cells. And number three, mesenchymal stem cells have the ability, at least in some situations, to repair secondary complications of diabetes. So we're going to discuss one of the secondary complications of diabetes, kidney failure, and we're going to discuss a paper in which kidney failure was treated in an animal model using mesenchymal stem cells. This paper is freely available online, and it was Togel et al. Was the name of the author. Now, what the investigators did is they took bone marrow cells and generated mesenchymal stem cells. And to make sure that they had mesenchymal stem cells, they induced them to differentiate, as you can see in the figure, into adipose cells or into bone cells, into osteocytes. And this is one of the characteristics for people that people use to know that the cells they're playing with really are mesenchymal cells. Now, these cells were subsequently used to treat a model of kidney failure. This model involves taking the rat and temporarily occluding the renal artery so that the kidney has no blood for 40 minutes. And then the occlusion is opened up, and what happens is ischemia reperfusion injury is called. Essentially, this is one of the models that's used to assess kidney failure. It's not diabetic kidney failure, but it's an animal model that mimics kidney failure. So when you administer 1 million mesenchymal cells systemically into the carotid artery, so they go all over the body, when you do this immediately after the kidney injury, and then you measure the serum creatinine being the marker of kidney injury, you can see that at day one, there's no difference between the creatinine in the rats that received stem cells, so that's filled in the black bars, and the control animals. Now you can see on day 2 and day 3 there is a difference. The creatinine is lower in the rats that received the mesenchymal stem cells. Now you might ask, is this something specific to mesenchymal stem cells, or can any other cell mediate such a protective effect? So, in the next series of experiments, the authors compared, using the same model, kidney occlusion, they compared injection of 1 million mesenchymal stem cells, which is in the middle, and 1 million fibroblasts, which are the control cells. And as you can see, the the mice that received fibroblasts, they had a high level of creatinine, similar to control mice that did not receive cells, whereas the mice that received mesenchymal stem cells had a reduction in the creatinine level. Now, next question that the investigators looked at is, serum creatinine is a functional marker, but what does the kidney actually look like? So they did what's called an injury score. They asked the pathologist to quantify the kidney damage, and as you can see in the figure, the kidney damage, the pathology score, the kidney injury score, is significantly lower in the rats that received the mesenchymal stem cells. Now, if there's healing going on, another question obviously is, what about proliferation? Is there multiplication going on? As the cells are healing, are new ones being made? So the rats were assessed after the stem cell therapy or controls for expression in the kidney of proliferating cell nuclear antigen, PCNA. And as you can see, the rats that received mesenchymal stem cells had a higher PCNA score. Part of the problem in kidney failure and in kidney damage is apoptosis, which is associated with acute tubular necrosis. Despite the word necrosis, it's actually apoptosis that's occurring. And as you can see in this figure, the apoptotic index, as measured by tunnel, is also reduced in the rats that received mesenchymal stem cells. Now, mechanistically, how can this be working? And to find this out, the investigators looked in the control and in the treated kidneys for expression of genes, inflammatory genes, TNF-alpha, interleukin-1, and interferon gamma, and also anti-inflammatory genes, the anti-inflammatory gene interleukin-10. And as you can see, as compared to control, the inflammatory cytokines were reduced, but interleukin-10 was very highly expressed. In fact, in the controls, there was no interleukin-10 expression, but in the animals that received mesenchymal stem cells, there was a higher level of interleukin-10 being expressed at the gene level. So, this study provides some indication, number one, that the mesenchymal stem cells can be introduced intravenously and still mediate their effect. Number two, this study shows that there is an effect in terms of repairing the kidney. Now mind you, this is an acute injury, and the cells were administered immediately after the injury, or in some other experiments which we didn't talk about, but they're in the paper, they were administered after the injury, 1 day after the injury. How well does this relate to the clinical situation? Well, in terms of diabetes, a little different, because in diabetes you have a much more chronic destruction of the kidney. It does relate, however, to situations like organ transplant, where an organ transplant, literally you do clamp the kidney because you're transplanting it and you have this ischemic perfusion injury. So one of the interesting uses would be use of mesenchymal stem cells to actually protect organs during transplantation. But again, getting back to diabetes, the study provides some indication that, at least in animals, the mesenchymal cells have some kidney protective effects. There's a lot of studies being performed on this, and you can find a lot more on PubMed.com. Thank you very much.